So the Biomarker means Biomarker of T cell activity in tumor and immunotherapy response. We are in the field of nuclear medicine and nuclear imaging, and we try to develop new biomarker to assess the response of immunotherapy, which are new therapies that are very efficient now to treat cancer. So the objective of this project was to develop new pharmacoimaging biomarker to evaluate T cell activity in tumors and identify also new biomarkers to predict the response to immunotherapies. Here, show you a small cartoon for the clinical context. When you have a tumor, you have to measure the immune biomarkers. It could be the T cells, like the CD8 cells, or it could be expression of a specific target, here PD-L1, but there are many others. And based on this immune biomarker, you can decide to use different treatments. Nivolumab is one of them. But in some case, you don't have the expression of the targets, or the patient are not respond to the therapy. And you need new biomarker, new tools. And imaging could be one of them to predict and also to monitor the efficiency of the treatment all along the disease and the development of the disease or the cure. Here you have a patient that has a tumor on the left with a specific imaging of the tumor with the T cells, and this patient has also a tumor, but there is no image of the biomarker. And this is the kind of tools we want to develop as well for new targets. We have 3 work packages in the program. The first one was to validate our strategy, the chemical strategy, the models, everything that has been used. And for that, we use the CD8 biomarker, which is well known already in clinic, to be used as a proof of concept to validate all the strategy. Then the work package 2 was more focused on the clinic to identify new biomarker based on 2 cohorts, one in the lung, one in the colon cancer, to identify this new immunotherapy response biomarker. And the first work package was to apply the strategy that has been developed in the first one to these new biomarkers. Some key figures. So the program has been launched in 2018. There was 6 partners, 6 funded positions. It's a global budget, including Diaclone 1.6 million, with the support of the ANA ISITE and the FIDER program. And it is all coordinated by PharmImage, and we meet every 2 weeks to push forward this program as much as we can. So the first work package with these 4 partners that have been involved was to develop, as I say, the CD8 imaging agents proof of concept. So starting from an anticore that has been developed initially by Diaclone, we cut it in small pieces. So with the antibodies, we can work different fragments. One is named FAB, the other one is named CFV. Well, doesn't matter too much the name. It's a small piece of antibodies, which are more efficient to be used in imaging. And then we engineer also these fragments by adding a cysteine, which helps us for the nuclear imaging, because we can really target the isotope where we want. So this is the first generation we developed. This is very classical. We call it a random conjugation. We take the fragment and we just apply the chemistry, and the NONAGA or the chelating agents per se will be attached to the fragment at different leasing position, but we cannot really control it. We had 4 fragments at the beginning, and we select the best among the 4 for the next generation, next strategy. So the second generation is more site-specific. This is where we add a cysteine to be more reproducible and define a better imaging agents. So the first strategy was to apply 1 chelator per fragment. This is a common strategy, and it has been well done. And the second one was to apply an innovative strategy developed by the ICMUN lab, what we call the tetrazine platform. And this technology allows us to attach 2 chelating agents to the fragments. So here we have, again, the same fragments. We have a linker, and with this linker, we can add the tetrazine platform, and at the ends, obtain a fragment with 2 NONAGA, so 2 chelating agents, what we call DNODAGA. So we have two chelators, so we have a better specific activity, we have better radioactivity to be imaged, and have a better contrast at the end. So then we have to screen between two different fragments, so either the single node-AGA or the G-node-AGA. We had different technology to screen it, to decide which one was the best, and if we improve by using the tetrazine platform, if we improve our fragments. We use the recombinant, we use the octet technology, which helps us to determine the affinity on a recombinant protein. And then we use flow cytometry to validate the affinity on CD8 cells or T cells or on human blood cells. And the affinity was more or less in the same range on the recombinant protein. And by flow cytometry, I won't go into details, this is the blue line that you can observe there is a shift. And so we observe, again, a good affinity, which was in the range of the initial antibody that we have used from Diaclone. And again, we observe the blue good affinity in the global human blood cells. Then we have to do the radiolabeling itself. So antiscalating agents. The purpose of Antiscalating agent is to be a cage for the isotope. Here is the gallium-68 that we have used, which is an isotope that has a half-life of 1 hour. So you have to be very fast. Basically, you use an automatic system. I don't know why the picture is not complete, but it has been cut. You have this automatic system that helps you to do the synthesis. And then we do the purification and we measure the specific activity. And as you can see, we go from 0.8 to 1.4, 1 to 1.4. So we improve the specific activity by adding 2 gallium-68 isotope, which was expected. And then we do the purification, so it's just a control, and we had a good purity of the compounds. So the next step was to validate, again, the affinity of this compound after the radiolabeling. So we did an immunoreactivity or an affinity assay for both of these compounds. And again, we were more or less in the same range. We had little loss of affinity with the D-node-AGA, but it remains still very good with 20 nanomolar. And then comes the in vivo experiments. So here it's more complex models. As the CD8 antibody that we use from the start recognize only the human CD8 and not the mouse CD8, we have to use the humanized mice. So for that, we purchased them from Oncodesign, which is one of the expertise of the company. And we develop a first model where we treat the mice first with Ramos tumor cells that was injected IV, then with human blood cells, and we treat them with blinatumomab, which is known to recruit more human cells to the site of the tumor. And we perform imaging and ex vivo analysis to collect after on the spleen and bone marrow at the end of the experiments. And here actually the pictures show that the level of Ramos cells and CD8 that we observe in these models was too low. It was in the range of 2 to 4%, which is not enough for imaging to be able to detect. So this model actually failed, and we had to look for another in vivo model to pursue to validate our imaging agents. And it was the purpose of this work package 1, to validate the whole global strategy. So we went back, and we screened different models that we had in our biobank. We, from our Oncodesign, share some samples from different humanized mouse models. And we select OVCAR-3, which is an ovarian cancer model. And here you can see on the left image, you have a control, which is an amygdala, which is positive for CD8. And then you have this ovarian cancer, which was also positive, which is the brown pitch, which brown set in the image. And then on the autoradiography is you take the same kind of samples and you bind your radiolabeled compound, so the D-node-AGA, the radiolabeled D-node-AGA. And you can see on the sample that has score 3, so positive for CD8, you have a positive signal on the right panel. And on the panel below, you have no positive signal or very weak signal, which was expected as a CD8, which is the sample was negative for CD8. So now we have validated, at least in vitro, that we have binding of our molecules on the samples. So now the next step is to do the in vivo imaging of this with these models, and the study is ongoing, and we expect the results in December, so in a few weeks from now. So the global strategy is more or less validated, and now we have to identify new biomarker, and this part of the work is done by the clinical group at the CHR in Besançon or at the CHFL. So from their clinical cohort, so lung cancer or colorectal cancer, they have different strategy. One is using RNA sequencing, the other one is using flow cytometry and comparing immune cells or targets within the tumor versus peripheral blood to get very specific signal from the tumor. They have globally identified 14 candidates. One is now validated and 2 are under investigation. What we are looking for is a biomarker that is highly expressed on the cell surface, because we need an imaging agent that will bind, that will not enter to the cells, so this is what we need. We need, of course, a tumor-specific expression, limited expression in normal tissue, and, of course, correlate with immunotherapy response. So this is the first target that has been identified. We have a high expression in cancer unrelated stroma on the upper left. We have a low expression on tumor normal cells. We have a high expression that is correlated to immunotherapy. I won't describe the graph in detail, but the purple column means that it's correlated, it's well expressed when the patients are treated with immunotherapy. And this expression correlates with bad prognosis in patients that is treated with this kind of therapy. So now we move this target to the work package 3. Currently, the diaclone is producing the antibody and then the scFv, and we will decline the same process that we are validating. Conclusion, we have now selected the best CD8 fragment. We have validated the multiple chemical strategy and notably the tetrazine approach. We have selected the in vivo models. In the second work package, we have now one out of the 3 targets that we would like to validate. And we really consider that this project so far is quite successful and combining, multidisciplinary know-how from chemistry to clinic, different partners from Bourgogne-Franche-Comté, and we hope to valorize that with the novel IP, scientific publication, and of course new commercial opportunities for diaclones.
Thank you very much for this very, very nice presentation.
And in blue, that's the people that are funded by.
Okay.
Thank you so much, Cyril, for this great presentation. So maybe you can talk about this new candidate. So obviously that must be a membrane-bound protein, so that you are targeting, no?
Yeah.
The targets that. So amongst the 14 that have been discovered from the CGFL, how many are putative membrane-bound proteins?
Sorry, I didn't get your exact question.
Among the 14 candidates
Yes.
that you presented as discriminating a tumor to a normal cell, how many proteins are membrane-bound among these targets?
Are membrane CART?
Membrane-bound protein.
Membrane protein. So actually, the 14 are membrane protein.
14 of them.
All
of them.
Yeah, there was more than 100 genes on the front, and the 14 were membrane. That was the first screening, and then it was more. The issue was more the level of expression and the selectivity of expression between tumors and normal tissues. We need to have very good separation of expression.
And with respect to antibody production, because you want to target and you're going to raise antibodies against these proteins.
Yeah.
So are you considering that maybe approaches that would keep the glycosylation or the post-translational modifications of these proteins would be important, because the usual way of generating antibodies is not, is not compatible with this?
No, here we clearly rely on diaclone expertise to validate this expertise and the strategy. They will not overexpress. The goal is to not overexpress the protein in the cells, but more to use a cell line or a cellular model that naturally express the cells for the screening.
Okay. Are there any some questions in the audience?
Are there any other questions?
So I think the new marker is highly confidential.
That's yeah, that's.
Can you tell us something about it or nothing?
It's a biomarker that is well expressed in the stroma, and as there will be IP, I cannot disclose the name.
Of course.
Of the program. And
Yeah, I was joking a little bit. But how many biomarkers do you plan to move to phase 3 or to in vivo imaging?
We hope to develop 3 total. That's the plan. Then it depends also if the models are available, because we have another target, for examples, that was very interesting, but it was related to lymphocyte B. But there is no good models, humanized model for lymphocyte B. And then it's a limitation. Yeah, big limitation. We may not be able to pursue this target due to the lack of models.
Okay, thank you very much. So if there's no urgent burning question, then maybe we should switch to the next speaker. Thank you again, Cyril.